Buongiorno everyone. My name is Iva Aleksic, and today I will briefly represent you my results from my first year of PhD studies. Parkinson's disease is typically considered as a chronic progressive neurodegenerative movement disorder. It is characterized by severe motor symptoms that are developed as a consequence of the degeneration of dopaminergic neurons in the substantia nigra. Dopamine is important neurotransmitter in the brain, but it is also identified as important molecule that bridging central nervous system and innate immune system. Further literature data that support this immunomodulatory function of dopamine are that dopaminergic receptors are expressed on the different immune cells, that there is specific endogenous transport system in leukocytes, and then there is endogenous synthesis in leukocytes of this catecholamine. Dopamine signals via 5 different dopaminergic receptors that are divided into different families based on pharmacological features and mechanism of action. The one-like family of receptors actually activate adenyl cyclase, and the 2-like receptor inhibit the same enzyme, and this is the way how they are control increase or decrease of secondary messenger molecule. Along with the neuroinflammatory process, the similar changes are observed in the periphery of immune system, and literature data support this, said that CD4 positive T cells infiltrate post-mortem human specimens, that there is decrease of CD4-CD8 ratio, and among CD4 positive T cells we have a decreased percentage of naive and regulatory T cells and increased percentage of memory T cells. Also, there is increased ratio of pro-inflammatory versus anti-inflammatory cytokines that these cells produce. All these observations said and suggest that there is important immunological mechanism that is involved in Parkinson's disease progression. In peripheral blood, naive CD4 positive T cells are able to differentiate into 4 different major lineages, and this process is led by the cytokines present in microsurrounding environment and transcriptional factors that are expressed in these cells. These cells are also divided by profile of cytokines that they are produced. We have pro-inflammatory Th1 and Th17 and anti-inflammatory Th2 subset. If we put this subset in the context of neuroinflammation in Parkinson's disease process, we can see that Th1 and Th17 cells are able to ingress into the central nervous system, change phenotype of microglial cells to pro-inflammatory. These cells then produce free radicals and pro-inflammatory cytokines and involved in neurotoxicity. On the other hand, we have Th2 cells that are able to upregulate neurotrophic factor and are involved in neuroprotection. T regulatory cells are also important key players in this process because they are able to suppress this pro-inflammatory response. Bearing all of this in our mind, aims of our study were to characterize a population of CD4 positive T helper lymphocytes with a special attention to Th1 and Th2 subset in the peripheral blood of healthy subjects and patients, and we were especially interested in pattern of expression of dopaminergic receptors on these cells. In our study, we involved peripheral venous blood and of healthy volunteers that were age-matched control of Parkinson's disease patient. We obtained leukocyte formula from the hospital and other methods that we use, and we save our samples for pharmacogenetics study for evaluation of cytokine and dopamine levels from plasma. We also isolate peripheral blood mononuclear cells for PCR and HPLC method, and a major method that we use and that I'm going to represent our data today is flow cytometry, which allow us to identify frequency of different TH subset and expression of dopaminergic receptors. Data of expression of dopaminergic receptor on CD4 positive cells and also subsets are not very well present in literature. And first I will speak only about controls. And this leukocyte formula actually says that in controls, all blood counts of cells were in the range of reference values. Gating strategy that we use is actually on B-parametric dot plot, SSC versus FSC, and we gated only lymphocytes. On these cells, we were able by single parametric histogram picture to represent only CD4 positive T cells, and on these cells we identify dopaminergic receptor regarding the negative control. Similar gating strategy was used for Th1 and Th2 subset cells with this additional step where we had the expression of two different chemokine receptors that are characteristic for CCR4 for Th2 subset and CXCR3 for Th1 subset, and we were also able to identify dopaminergic receptor on these cells. Briefly, what we got is that among CD4 positive T cells, we have expression of, we have a Th1 subset in 27% and Th2 subset in 15% of cells. And when we look for the expression of dopaminergic receptors on all CD4 positive T cells from whole blood of healthy control, we can say that these receptors are present, that all 5 receptors are present, that D1-like receptors are highly expressed compared to D2-like receptors, and that among D2-like receptors, D2-like D2 receptor is lower expressed than D3 receptor. When we go through Th1 and Th2 subset, also in healthy controls, we can see that comparing D1-like and D2 receptors, we also have a higher expression of D1-like receptor, and among D2-like receptor, we have a difference between D2 and D4. If we only look for the expression of individual dopaminergic receptors between these two Th1, Th2 subset, we can say that D1 receptors are highly expressed in Th2 subset. When we compare patients and control samples, we express these results as a percentage of cells, and among these we can see that, for example, D4 receptor is higher expressed in patients, and between absolute numbers we can say that there is a difference between D2 and D3 like D2 and D3 receptors that are lower presented on patient group. Further on, when we go to through Th1 subset, we can see that there is no statistical difference among percentages or absolute number of these cells, but the trend remain the same, and between Th2 subset, we can also say that there is no statistical significance between percentages or among absolute number of these cells, but the trend also was kept. So in conclusions, we can say that human CD4 positive lymphocytes express all 5 subtypes of dopaminergic receptor with the distinct patterns between Th1 and Th2, that the higher frequency of D1 that we show the higher frequency of D1-like receptors compared to D2-like receptors in different Th subset, and that when we compared controls and patients, the expression of the dopaminergic receptor was quite similar with very few statistical difference. At the end, I would like to say thank you to all of my group and colleagues that support me for these results and in my work. Thank you so much.